Hi friends, good evening. How are you? Hope all are fine. Welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Today we shall discuss about actin filaments. Actin filaments are the principal component of cytoskeleton. These filaments consist of a globular protein known as actin. Actin can exist in two forms. One is G-actin, that is known as globular actin. Other is F-actin, that means filamentous actin. So now let us discuss about actin filaments. Actin filaments are also known as microfilaments. These actin filaments are thin, flexible fiber. The diameter is about 7 nanometer and the filaments can be up to several micrometer in length. This is a actin filament. The diameter of the actin filament is about 7 nanometer and the length of the actin filament can be up to several micrometer. These actin filaments can be organized into bundles or network. Actin filaments are dynamic structure, that means they undergo continual assembly and disassembly within the cell. The actin filaments are abundant beneath the plasma membrane where it forms a network. This network provides mechanical support. It also determines cell shape and also allows the movement of cell surface that enable the cells to migrate, to engulf and to divide. Assembly and disassembly of actin filaments. The actin filaments consist of a type of globular protein that is known as actin. This actin, which is also known as G-actin, is a single polypeptide chain. Molecular weight is 43 kilodalton and the polypeptide chain contains 375 amino acids. So this G-actin is also known as actin monomer. The actin monomer, this is a actin monomer. The actin monomers have tight binding site that mediate head-to-tail interaction with two actin monomer. This binding of actin monomer with two actin monomer leads to the process of polymerization. Polymerization of actin filament is a process in which actin monomers are polymerized into filament. And in this filament, each monomer is rotated by 166 degree in the filament, so it appears like a double-stranded helix. These actin filaments are polar structure, that means they have distinct plus end and minus end. The plus end elongate 5 to 10 times faster than the minus end. This is the plus end and this is the minus end. The plus end of the actin filament elongate 5 to 10 times greater than the minus end. So what are the steps in actin polymerization? First step is known as nucleation. Nucleation is the formation of aggregates consisting of three actin monomers. Here you see three actin monomers have combined with each other to form aggregates. Second step is growth of the actin filament by the reversible addition of monomer to both ends. So polymerization occurs through the growth of the filament by the reversible addition of monomer to both ends. This is a actin filament, this is the plus end and this is the minus end. Actin monomers are added both to the plus end and also to the minus end, but the plus end elongate 5 to 10 times faster than the minus end. This actin filament, it consists of two helical aggregates of G-actin that are twisted around each other, having 13.5 subunits per turn. So this is the structure of actin filaments. This actin filament consists of two helical aggregates of G-actin that are twisted around each other, and this actin filament has 13.5 subunits per turn. The actin monomers also bind ATP, which is hydrolyzed following the assembly of the filament. ATP is not required for polymerization, but the actin monomer, which is bound to ATP, it polymerizes readily than the actin monomer that is bound to ADP. So the binding of ATP and the hydrolysis of ATP play a key role in regulating the assembly of microfilament or actin filament. This also regulates the dynamic behavior of the actin filament. Actin polymerization is a reversible process. So the actin filaments can be depolymerized by the dissociation of the actin monomer. So the actin filament undergo breakdown, and there is an apparent equilibrium that exists between actin monomer and actin filament, and this equilibrium is dependent on the concentration of actin monomer. The rate of incorporation of actin monomer into the filament is proportional to the concentration of the monomer. Greater the concentration of the monomer, greater will be the rate of incorporation of the monomer into the actin filament. So, there is a critical concentration. What is critical concentration? At critical concentration, the rate of incorporation of actin monomer is equal to the rate of dissociation of monomer from the actin filament. And at critical concentration, there is an equilibrium between actin monomer and the actin filament. Then the actin filaments are polar structure. That means the actin filament has two distinct ends. One is plus end, other is minus end. And the plus end elongate 5 to 10 times faster than the minus end. And because the ATP actin dissociate less readily as compared to ADP actin, so there is a difference in the critical concentration required for incorporation of actin monomer into the plus end and the minus end. And this difference in critical concentration result in treadmilling. What is treadmilling? It is a dynamic behavior. Treadmilling is a dynamic behavior in which one end of the actin filament grow and the other end shrink. So, due to this treadmilling, a section of the actin filament seemingly moves. Next, assembly and disassembly of actin filament. Assembly and disassembly of actin filament is necessary for remodeling of the cytoskeleton. And assembly and disassembly is regulated by various actin binding proteins. Various actin binding proteins like cofilin, profilin, and ARP2 by 3 proteins. All of these proteins play a regulatory role in the assembly and disassembly of actin filament. First is cofilin. Cofilin is a actin binding protein that enhances the dissociation of actin monomer from the actin filament. This cofilin preferentially binds to ADP actin, so it sequesters actin in the form of ADP actin. Another protein is profilin. Profilin, it promotes or it stimulates the incorporation of actin monomer into the actin filament. Profilin, it promotes the exchange of bound ATP for ATP, so this results in the formation of ATP actin monomer that dissociate from cofilin, so it becomes available for incorporation into the actin filament. Other protein is ARP2 by 3 proteins. These proteins act as nucleation sites for initiation of assembly of new actin filaments. So cofilin, profilin and ARP2 by 3 proteins, they act together to promote the rapid turnover of actin filaments that result in cytoskeleton remodeling, which is necessary for cell movement and change in cell shape. Friends, this is about actin filament. Please share, like and subscribe this video, and also please give your comments so that I will be able to improve my performance. Thank you. Bye bye.